Good morning, everyone.
Good morning.
Yeah, I think there are 5 sections for this morning, so you guys, I guess, is tired. So, last week we talked about types of adaptive immune response. Now we know there are 2 types of adaptive immune responses called humoral immunity and cell-mediated immunity. Humoral immunity is mediated by antibody, and a cell-mediated immunity, also called cellular immunity, is mediated by T lymphocyte. So from innate immunity to adaptive immunity, different components of the cell or molecule involved in immune response. In innate immunity, several kinds of the cells include macrophage, natural killer cells, and some of the molecules include complement system and C reaction protein. And in adaptive immunity, there are 2 types of the lymphocyte, T lymphocyte and the B lymphocyte, involved in the immune response. And also, its product we call antibody, which secreted by plasma cell, derived from B lymphocyte. We call antibody. We will discuss these contents in detail next. So, types of adaptive immunity, humoral and cell-mediated immunity, microbes binding from B lymphocyte. We call antibody. We will discuss these contents in detail next. So, types of adaptive immunity, humoral and cell-mediated immunity, microbes binding B lymphocyte, B cell receptor recognize the antigen of the microbe. And also, B lymphocytes are activated, proliferate, and differentiate to plasma cell to produce or circulate antibody, block the microbes, or we sometimes call pathogen, to infect other new cell. This procedure we call neutralization. Block infection and eliminate extracellular microbes through the other mechanism or the other procedure, something like complement system activation. We will talk about later. And antibody binding to the effector cell, which located on macrophage and natural killer cell, go through the ADCC or phagocytosis, finally to eliminate the pathogen. Cell-mediated immunity, T cell recognition of the antigen. This is the key, or we call core procedure of the adaptive immune response. APC, antigen presenting cell, present the antigen for T cell recognition. Then T cell were activated, proliferate, and differentiate to the different kinds of the effector cell, include T helper 1, T helper 2, or finally to CTL. So activate T lymphocyte produce cytokine, further activate macrophage. And the macrophage is one of the APC. They present antigen for T lymphocyte. And also, there are T cell receptor. T cell receptor recognize epitope of the antigen. Then activate macrophage to kill microbes by phagocytosis. Or intracellular microbes, some of the pathogens get into the inner cell. In this case, like the mutated cell or like the cell infected by the virus, these are target cells. And also, they are antigen-presenting cells. They present the intracellular protein or antigen in the cellular, and those endogenous antigen to the T lymphocyte. So this is a cytolytic T lymphocyte. And right now we know is a CD8 positive. CD8 positive. In this case, CTL, kill infected cell and eliminate reservoirs of the infection. There are several major features of the adaptive immune response. The first is specificity. That is a very good model for us to understand the specificity of the immune response, of the adaptive immune response. Vaccination. I guess all of the people sitting here got the vaccine, different kind of vaccine. That means immunization. After the vaccination, the human body or immune system will develop specific immunity against the specific pathogen infection. So immune responses are specific for distinct antigens. How? Why the recognition is specific? We talked about the clonal selection last week. Specific T lymphocyte or specific B lymphocyte. When these two groups of the T lymphocyte encounter a specific antigen, then this clone was selected. So, stimulate and response, this is a basic idea. Stimulation, response. For the immunology, stimulation is antigen stimulation. But for the other subject like drug or oxygen or nutrition, without nutrition is also one kind of stimulation. And the cell will respond, go through the signal transduction. In fact, for different portions of the single complex protein, polysaccharide, or other macromolecules, the parts of the such antigen that are specifically recognized by individual lymphocytes are called determinants, or sometimes we call epitope. This is the clonal selection. If you got shot against HIV, then the human body will develop the immunity against HIV infection, but could not defend the other virus, the other kind of virus or other pathogen to invade into the human body. So the adaptive immune response is specific. Second, the diversity. The reservoir of the T lymphocyte or the B lymphocyte could be reached to over 10 times 7 or more. During the development of the TCR or BCR, this is a T cell receptor or B cell receptor. The mature TCR or BCR occurred in the central immune organs. T cell developed in the thymus, and the B cell developed in the bone marrow. During the development, T cell receptor and the B cell receptor, the genes were expressed completely. And in this case, the gene from the germline rearrangement, so the product is quite different. In this case, this kind of the T cell receptor and the B cell receptor could recognize the various of the antigen. So this picture we call diversity. The total number of the antigenic specificities of the lymphocyte in an individual is extremely large. So this property of the lymphocyte repertoire is called diversity. In other words, there are many different clones of lymphocytes that differ in the structure of the antigen receptor, and therefore in the specificity for antigens, creating a total repertoire that is extremely diverse. And also there is a very good example or model for us to understand this picture: immunization. When you got the shot, the vaccine of the HBV, or got the shot for an HIV or other vaccine, so many different kind of the specific immune response form the diversity. The third feature is memory. Also, we can think about immunization of vaccine. Some vaccine could develop the lifetime immunity against the pathogen. Some of the vaccine need to be shot again and again. Just depends on the different time interval. Maybe, for example, for HBV vaccination, after the individual was born in a month, then got the first shot. 2 months later, the second shot. 6 months later, the third time shot. So, in this case, immune system will develop the memory. By the time the human body encounters the same antigen infection, then the immune system will develop more rapidly, more stronger immune response to against this specific pathogen. So exposure of the immune system to a foreign antigen enhances its ability to respond again to that antigen. Responses to second and the subsequent exposure to the same antigen we called secondary immune response, and are usually more rapid, larger, and often qualitatively different from the first or primary immune response to that antigen. We will have the picture to show these characteristics. So for individuals who never encounter or a particular antigen are said to be naive. Individuals who have responded to a microbial antigen and are protected from subsequent exposure to that microbe are said to be immune. The fourth is self-limitation. So we already talked about these features last week. So right now I just briefly review this picture, self-limitation. we call homeostasis. And this is the primary immune response. And this is the secondary immune response. More stronger, more rapidly. And then this is the primary immune response. Specialization. Immune system developed the optimized immune response against the same pathogen infection at different stage or different pathogen infection, either by humoral immunity or by cell immunity response. Just depends on different kind of the pathogen infection or same pathogen infection at different stage, like the outer extracellular or inner cellular. Non-reactivity to the cell. This is the major feature of the immune system. During the lymphocyte development in the central immune organs, T lymphocyte or B lymphocyte will develop the immune tolerance. This meaning, during the recognition, those clones of the T or B lymphocyte react to the self substances. Those clones will be deleted by apoptosis. That means tolerance to the self and against to the non-self in most of the case. But sometimes people will show the tolerance to the pathogen. That means the infection was established. Or sometimes the immune system against its own substances. That means autoimmune response. That means immune system against itself. So specificity, diversity, memory, specialization, self-limitation, and non-reactivity to self is the sixth feature of the immune responses. So cellular components of the adaptive immune system. Two types of the lymphocyte involved in the adaptive immune system. One is T lymphocyte, the other is B lymphocyte. B lymphocytes are the only cells capable of producing antibody. Usually they recognize extracellular antigens. The transportation of the molecule is quite difficult to pass the cell membrane. In this case, some of the molecules try to get into the inner cell, need receptor to help those molecules transmembrane transportation. So antibody could not get inner the cell. In this case, how immune system eliminate the virus in the cell? So usually antibody recognize extracellular antigen. And the T lymphocyte, the cells of cell-mediated immunity recognize the antigens of intracellular microbes and function to destroy these microbes or the infected cell by produce something like the enzyme or produce something like a perforin, or let me we show the cytotoxic, cellular cytotoxic to its targeted cell. Or sometimes T cell, the molecule located on the T lymphocyte is present and binding to the receptor or the ligands of the target cell, then the apoptosis pathway is activated. T lymphocytes have restricted specificity for antigen. This feature we call major histocompatibility complex restriction. CD4 positive T lymphocytes recognize the antigen presented by antigen presenting cell. These antigen form the complex with the MHC class II molecules. And CD8 positive T lymphocyte recognize the antigen presented by APC, which form the complex with the MHC class I molecule. So this is we call MHC complex. Different effector cell. The biological function or the immunological function is different. So the initiation and development of the adaptive immune response require that antigen be captured and displayed to a specific lymphocyte. The cells that serve this role are called antigen-presenting cells. The activation of lymphocyte by antigen leads to the generation of numerous mechanisms that function to eliminate the antigen. Antigen elimination often requires the participation of the cells called effector cell. So we call the cell number of the immune cell or of the immune system is a fundamental of the immune response. You need to expansion of the lymphocyte. How lymphocyte could be expanded just upon to the activation. What kind of substance could activate lymphocyte? Antigen or some or other substances we will discuss in the chapter of the antigen. Activated T lymphocyte, mononuclear phagocyte, and other leukocytes function as effector cell in different immune response to eliminate different kind of the pathogen. Either is extracellular or intracellular of the cell. B lymphocyte binding to the microbe, binding to the antigen of the microbe. That means we call the neutralization of the microbe. Block the microbe to infect new cell. And T lymphocyte recognize the antigen presented by antigen presenting cell. Then T cell were activated. Activated T lymphocyte produce different kind of the cytokine. For example, interferon gamma, interleukin 2, or TNF alpha. To active like the TNF gamma activate of the macrophage to promote its phagocytosis, to promote the antigen presenting of the macrophage. And also, activated T lymphocyte produce some of the cytokine related to the inflammation, like interleukin 1, interleukin 6, interleukin 8, and also produce interleukin 10. Interleukin 10 is an inhibition of the inflammation. This is the regulation of the immune response or regulation of the inflammation. And also, activated T lymphocyte produce cytokine, this kind of cytokine proliferate, activated T lymphocyte proliferation and go through the differentiation to differentiate different group of the effector cell. And cytolytic T lymphocyte show its cytotoxic to the infected cell and kill the infected cell. And also, activated T lymphocyte produce interferon gamma to activate natural killer cell to kill the infected cell or cancer cell. So we just divide the adaptive immune response. This processing will divide into 3 stages. The first stage is recognition. The second stage is activation. The third stage is the effector phase after the differentiation of the lymphocyte. And finally, this immune response waned by time, just go back to its resting status. We call self-limitation, or we call homeostasis. The naive T lymphocyte and naive B lymphocyte receive the antigen stimulation, become activated, then go through clonal expansion to differentiate different effector cell. For B cell, produce antibody. For T lymphocyte, T cell, either differentiate into Th1 or Th2. Th2 means T helper cell 2, type 2, help B lymphocyte to produce antibody. And go through the different kind of the mechanism or procedure to eliminate the pathogen, then finally go through the homeostasis. Most of the effector cell die by apoptosis. Only a small part of the effector cell differentiate into the memory cell. This is why immune system, the immune response show one of the feature we call memory. So the first stage is recognition of antigen. How T cell or B cell could recognize antigen? In this case, we need to understand or learn some of the specific molecular located on the B cell, on the surface of the T cell or B cell. The first is T cell receptor or B cell receptor. So the first stage is recognition. And for the T cell, recognize the antigen from the APC, antigen presenting cell. B cell could recognize the antigen directly. And also, B cell is one of the antigen presenting cell. So, every individual possesses numerous clonal derived lymphocytes. Different kind of lymphocytes derived from the stem cell in bone marrow. Each clone having arisen from a single precursor and being capable of recognizing and responding to a distinct antigenic determinant. And when the antigen enters, it selects a specific pre-existing clone and activates it. This fundamental concept is called clonal selection hypothesis. So, if you understand the clonal selection, then it will help you to understand why immune response is specific. The second stage, activation of the lymphocyte. So we need to learn about cell surface marker or cell surface molecule located on the surface of the T lymphocyte or the B lymphocyte. How many molecules involved in the procedure of the activation of the lymphocyte? Some of the specific antibody were used in the clinical practice just upon the activation of the lymphocyte. Sometimes we need to block it, that means inhibit its activation. Sometimes we need to promote its activation. Just depends on what kind of purpose in clinical practice. So, for the T lymphocyte activation, need two signals. One is signal 1. One is antigen signal. Second is signal derived from the some local receptor. If without second signal, then T lymphocyte will go through the apoptosis or go through the allergy. We will discuss this procedure in detail later. So the two signal requirement for lymphocyte activation. The third stage is effector phase of immune response to elimination of the antigen. So how many effector cells involved in the elimination of the antigen? Communication between innate and adaptive immunity. Innate immunity and adaptive immunity is closely related. Adaptive immunity is initiated by innate immune cell, like macrophage, like dendritic cell. And also, innate immune cell express co-stimulating molecules or components to help the activation of the adaptive immune response. Some of the cytokines produced by innate immune cell, like natural killer cell, like the macrophage, those are cytokines produced by NK cell or macrophage, will help to direct differentiation of the T lymphocyte. So, antigen presenting cell antigen presenting cell present antigen to T lymphocyte. T lymphocyte differentiate to Th1 or Th2. How T cell could be differentiated to Th1 or Th2? Just depends on the cytokine produced by antigen presenting cell. For example, interferon gamma help T cell differentiate to Th1. Interleukin-4 help to T cell to differentiate to the Th1. The shift between Th1 and the Th2 means the types of the adaptive immunity is different. Either cellular immune response is dominant, or humoral immune response is dominant. So the innate immunity and the adaptive immunity is closely related. We will talk about later about the innate immunity and the adaptive immunity. By that time, it's easy for you to understand. Right now, you will feel confused. So innate and adaptive immune mechanism empower each other. Either one does not function well alone. Macrophage capture the pathogen, go through the phagocytosis, and also produce co-stimulator and cytokine. And cytokine unleash antigen presenting cell, present the antigen for naive T cell, and activation and differentiation depends on its differentiation depends on the different cytokine. And microbes will bind by the antibody. Antigen form the IC complex. We call it IC. What is immune complex? Antigen, antibody bind together, form immune complex. Immune complex trigger the activation of the complement system. We call this procedure is classical pathway. And in this case, complement system activate, then eliminate the pathogen by MAC, we call it membrane attack complex, to destroy the pathogen or infected cell. And also there are so many receptors located on the cell surface of the B lymphocyte. CR2, that means the complement receptor. Complement could bind into the receptor, then B cell proliferate and differentiate, mediated the humoral immunity. And finally, after the immune system eliminates the pathogen, the immune system returns to its basal resting state. We call it homeostasis. And some of the immune dysfunction, allergic and asthma, that means overreaction, or we call hypersensitivity. Allergic, is there anybody allergic to the seafood? Oh, sorry. And is anybody allergic to some of the medicine, antibiotics, like the ampicillin? No, that's good news, yeah. And graft rejection, that means also it's an overreaction. In this case, sometimes we use some of the inhibitor of the immune response. That means the human body shows the tolerance to the donor. And autoimmune disease also means something wrong with the recognition between self and the non-self. And the immune cell shows the reactivity to its own substances. This will develop the autoimmune disease. And some of the people show the immunodeficiency, like acquired immunodeficiency syndrome, we call AIDS. So allergy and asthma are results of the inappropriate immune responses often to common antigens such as plant pollen, food, and animal dander. And the cell organ transplantation. In some cases, the transplantation of a cell or an organ from another individual. So before we perform the cell or organ transplantation, you need to check like the blood perfusion, you need to check the ABO blood type. Is everybody know your own type blood? Yeah, okay. And for the organ transplantation, we need to check the HLA or MHC. HLA in human, MHC in human being we call HLA. Although an organ from another individual, although viewed by the immune system as a foreign invasion, may be the only possible treatment for disease, because the immune system will attack and reject any transplanted organ that it does not recognize as self. It is a serious barrier to these potential life-saving treatments. So we will talk about the immune response of the transplantation later. And autoimmune disease, yeah. In certain individuals, the immune system malfunction by losing its sense of self and non-self. So the key concept of the immune response is recognition. Distinguish self and the non-self. If there's something wrong with this procedure, then some related immune disease will develop. Either it's a higher or overreaction, or show the deficiency, or show the tolerance. So immunodeficiency is a good example for acquired immunodeficiency syndrome. We call it AIDS. So this is the overview of the immunology. I hope you will build up the major concept related immunology. Then in a later chapter, we will discuss in detail for the antigen, antibody. So now we know the immune system. We know the components of the immune system, immune organ and tissue. How many immune organ or tissue of the immune system? We now know the central and peripheral organ. Then, how many immune cells? Antigen presenting cell, lymphocyte, innate immune cell. And molecules include soluble molecules and membrane molecules. How many molecules include in soluble, like antibody, cytokine, and the complement system? And how many molecules is divided into the cell membrane molecules, like T cell receptor, B cell receptor, and some of the co-stimulator. This kind of the molecule is very important. And some of the molecules show its biological function by adhesion. So we call those molecules is adhesion molecules. We will discuss in detail on the later chapter. Okay, we will take 10 minutes break.